Hello, my name is Tavia Hall, and today I'm going to be talking about immunoglobulin proteins, and in particular, two that are very important for various developmental processes, imagin and basogen. My objectives today are to discuss the overall structure of immunoglobulin proteins, to also go through published literature that discusses the role of these proteins in regulatory mechanisms and specifically the importance of imagin and basogen in these processes. Finally, I would like to discuss further research that needs to be done in order to characterize the role of these genes in these cell processes. To understand immunoglobulin proteins, first we need to understand a basic protein structure. So there are four levels of protein structure. The most basic is primary structure. A primary structure of a protein refers to the simple sequence of amino acids as a transcript is translated into a polypeptide. Now the word polypeptide refers to poly. If you break the word down, poly means several, and peptide refers to the building blocks, the amino acids that are put together to construct a protein. The secondary structure is influenced by hydrogen bonding between the amino acids of the primary structure. Hydrogen bonding occurs from attraction between polar molecules, and these attractions result in one of two or a combination of two structures, two main structures, alpha helices, which can be described as a spring-like coil, and also beta pleated sheets, which are kind of folded fan-like, one goes up one way and turns around and continues back in the other direction. And this continues to make a long flat-like sheet, which is why it's called a beta pleated sheets. If you imagine it, it's like a big square that is composed of several different pleats, a fan fold, if you will. Tertiary structure refers to the hydrophobic or hydrophilic interactions of the proteins and of the peptides, and also the R groups that are connected to the amino acids. The term R group refers to any kind of carbon-based group that is attached to amino acids. The basic structure of an amino acid is the same. However, there are differing R groups which dictates the reactivity and the binding that will occur with each amino acid. The overall aim of tertiary structure is to protect the hydrophobic parts of the protein, the parts that are not likely to interact with the aqueous environment in the cell, the water-based environment, and to place those R groups and those areas of the protein that will coexist with water towards the outside and basically reach the lowest energy conformation. The final level of protein structure is quaternary structure, and it involves interfolding of multiple proteins, multiple tertiary structures, including several different types of proteins. Now, this can be several copies of identical proteins. This can be several different proteins coming together, or it can be a combination thereof. But most proteins that are used for cellular processes in our body are quaternary proteins. Here this example shows hemoglobin, which is a quaternary protein that's involved in carrying oxygen through the circulatory system. So the immunoglobulin superfamily of proteins, they are transmembrane glycoproteins. Transmembrane glycoproteins. What does that mean exactly? Well, transmembrane refers to the placement of these proteins, and this indicates that they are within the membrane, the cell membrane. So they are actually one edge of a protein will reach towards the intracellular space inside the cell, and the other side of the protein will extend out into the extracellular space on the other side of the protein. So it kind of acts as a go-between through the plasma membrane. The word glycoprotein refers to a protein that is attached to a carbohydrate side chain. So glycoproteins are frequently found in the cell membrane, and glycoproteins are frequently found in the cell membrane. Immunoglobulins are also what we call cellular adhesion molecules, which is frequently abbreviated as a CAM or CAMs. Cellular adhesion molecules are exactly as their name describes them. They are molecules or proteins that are involved in binding of cells, so cell-to-cell interactions or cell to extracellular matrix interactions. The immunoglobulin superfamily has a very characteristic structure. They have anti-parallel beta-pleated sheets, which means that they are going opposite ways. It's almost a mirror image. And they are sandwiched between two other beta-pleated sheets for a total of 4. In the background protein, you can actually see an example of an immunoglobulin, and you can see 4 somewhat identical proteins that are kind of sandwiched together, but they are anti-parallel. They're not facing exactly the same way. They're mirror imaged almost. The immunoglobulins, they have a structure analogous to that of an antibody as well, which is really important, which plays a large role in its immune response, in its response in the body and its immune response. So here we see the antibody structure, and the structure of an antibody is Y-shaped, with the base of the Y known as the constant region, and the edges or the arms of the Y as the variable regions. Now all this means is that the variable regions or the variable segments is where their binding is going to occur. There will not be binding with any antigens on the constant in the constant domain. So we also see a difference between heavy chains and light chains. Now the heavy chains are continuous and are part of the constant domains, while the light chain are only included in the variable segments. One thing that's specific to Ig proteins is the presence of a disulfide bond, which can be seen in the middle and throughout the antibody. This disulfide bond creates a bridge between 2 different cytosines, which are amino acids, in this polypeptide, and they bind together, creating a bridge, and the excess protein in between those 2 cytosines forms a loop-like structure. It's this characteristic loop structure that immunoglobulin proteins are known for. So